Rocking M Studios and Mead Steel Education Productions present Wild World of Science, Genetic Engineering, with course instructor, Mr. Ronald Jowsey.
Greetings one and all, and welcome back. Today we are going to talk about the important process of genetic engineering. After this lesson, you should be able to define what genetic engineering is, list and describe the steps involved with genetic engineering, and give at least one example of how genetic engineering is helpful to humans. To guide us in our learning, we will be answering specific questions about genetic engineering by completing your genetic engineering worksheet. Let's get started, shall we? Genetic engineering is also known by other names. These include gene splicing, recombinant DNA, and genetic modification. Genetic engineering is combining DNA from two or more different organisms to create recombinant DNA. Recombinant DNA is DNA from two or more different organisms that has been combined. For example, if we take DNA from organism A and combine it with DNA from organism B, we create recombinant DNA. In this example, the gene that codes for glowing in the dark in jellyfish was combined with DNA from a cat to produce recombinant DNA that causes a cat to glow in the dark. Genetic engineering is possible because the genetic code is universal. This means that all living things have the same four possible DNA bases, A, T, C, and G. It is only the number and order of the DNA bases that makes one organism different from another organism. This brings us to the role that enzymes play in genetic engineering. We will be discussing two types of enzymes. The first enzyme that plays a role in genetic engineering is called a restriction enzyme. Restriction enzymes are used to cut DNA at specific locations. You should note that different kinds of restriction enzymes will cut DNA at different locations. The second type of enzyme involved in genetic engineering is called ligase. Ligase acts as a glue to put DNA segments back together. There are many different restriction enzymes, and each type of restriction enzyme cuts at a different sequence of DNA bases. Notice in the image that restriction enzyme 1 recognizes and cuts DNA at different base sequences than restriction enzymes 2 and 3. The base sequence, where a restriction enzyme cuts DNA, is known as the restriction site. When restriction enzymes cut the DNA, they often cut unevenly, leaving sticky ends. Step 1 in the process of genetic engineering is to use a specific restriction enzyme to cut DNA into fragments. Step 2 in the process is the addition of a DNA fragment cut from another source, using the same restriction enzyme. In Step 3, the two fragments stick together by complementary base pairing. In Step 4, DNA ligase pastes the sugar phosphate backbone together. Once DNA from two different organisms is combined, we have recombinant DNA. As a result of genetic engineering, bacteria cells can be factories that produce human insulin for diabetics. Bacteria are often used in genetic engineering because they have plasmids. Recall from previous lessons that plasmids are small circular pieces of DNA. Bacteria also reproduce asexually and produce genetically identical offspring, and bacteria produce large numbers of offspring quickly. Typical genetic engineering protocol in the manufacture of human insulin is as follows. In Step 1, a plasmid is isolated from a bacteria cell. In Step 2, the gene for human insulin is isolated from the DNA of a human cell. In Step 3, a specific restriction enzyme is used to cut the plasmid. In Step 4, the same restriction enzyme is used to cut the gene for human insulin from the human DNA strand. During Step 5, the human gene for insulin is combined with the plasmid DNA from the bacteria. During Step 6, Ligase is used to attach the plasmid and gene, forming a recombinant DNA plasmid. In step 7, the resulting recombinant DNA plasmid, composed of the bacteria DNA and the gene for human insulin, is placed back into a bacterium. During step 8, the recombinant bacteria is allowed to reproduce. Because bacteria reproduce asexually, every offspring produced by the recombinant bacteria will produce human insulin. And finally, during step 9, the insulin protein is isolated and collected from the cloned bacteria. The production of human insulin to treat diabetics is just one benefit of genetic engineering. Further examples include pest and disease-resistant plants, bacteria that clean toxic waste, protein that dissolves blood clots in heart attack therapy, growth hormone to treat stunted growth, and even acid-wash genes. That finishes our look at the process and benefits of genetic engineering. Be sure to turn in your worksheet when you've completed it. Until next time, be safe, make good choices, and I'll see you soon.